other guests that are in the channel. Let's see if I can get rid of this thing. All right. So we look at assembling macrocycles, and you can think of these assembling macrocycles as giving porous materials. So especially if they have a sizable hole in the center. And porous materials are a really exciting area. There's a lot of different strategies to making solids that have interior open spaces. The reason there's so many different strategies is because they have so many cool applications. They're great for separations, catalysis. We look at reactions and flow and diffusion in confined environments. Some of the things that we were, that we were quite excited about were this idea of being able to control the interior space to do catalysis. And so the host guest community has done phenomenal work in this area. Here's an older host from the standards group, where you can control the size and shape and functionality that point into that interior. And so we wanted to combine that type of control with an assembly that would give us a larger porous structure. And so like zeolites, we have tiny pores that range in the nanometer and sub-nanometer range, where we can look at containing materials, looking at flow in tiny spaces and reactions in tiny spaces. Our pores are very small and compared to what you would see from a moth. Those are giant from our perspective. COFs are newer type materials, covalent organic frameworks or porous organic polymers. I guess initially ours would fit into these molecularly porous materials. I guess since the time that we started, there's also been another name for these materials, kind of hydrogen bonded organic frameworks or HOFs. Some of our group interests have been in looking at frameworks that can polymerize. And for these materials, let's see if I can move this so I don't see, all I see is pictures here. Okay. So we made tetra amide system that pre-organized diacetylenes into columnar materials. And then when you heated them, these underwent a single crystal to single crystal transformation to make porous polydiacetylene nanofibers, which was really cool for us. We never able to get funding in this area, but we got some cool papers out and we're able to answer some neat questions. We're also interested in photocatalysis. Here's an earlier work where we used ruthenium complex with this pyridyl bisurea macrocycle to do Diels-Alder reaction, to catalyze the Diels-Alder reaction. We're moving into this, back into this area. Initially, we hadn't done that much work in the area because our cyclization conditions gave quite low yields. And if you want to screen a lot of reactions for catalysis, you have to have a large amount of your material. So we're still, I'll point out areas where we're working on that now. So when we started in these bisurea macrocycles, a little about 15 years ago, our design principle was very simple. So what we did was we used a urea, right? We were going to use a urea, but ureas are very good at doing gelating and assembly. And so we needed to control how they assembled. And so we do that by using two C-shaped spacers. These pre-organize the urea groups approximately perpendicular to the plane of the macrocycle. And under these conditions, they assemble really nicely in high fidelity to give you columnar structures. So depending on if these, the macrocycles themselves have a nice channel, have a nice hull, you'll get a straw structure with diameters ranging from 3 to about 9 angstroms in diameter, is the largest systems that we have. If they don't really have any kind of hull, then these are more like assembling cylinders, right, that wouldn't have a pore in those. And that's a kind of an example from these metaxylene ones. So they don't really have any channel, but things like this benzophenone or phenyl ether have a channel of about 5 angstroms in diameter. And our largest systems, this phenylethylamine, have a channel of about 9 angstroms in diameter. These materials, though, are assembled into crystals, and they're really homogeneous. And so there are advantages of using a homogeneous material to study flow and diffusion and to develop new techniques to look at confinement. And so I'm going to just highlight a few of these in my talk. We'll just look at some of our materials in terms of reactions, photooxidations, persistent radicals, and flow. We've done a little bit with in tiny channels. So this pyridol bacteria, this one assembled interdigitatedly, so not directly into columns. So that had a smaller hole, a channel of only 4 angstroms, 4.5 angstroms. And so this was nice to bind isoprene and could catalyze the UV-initiated polymerization of isoprene within the tiny channels. So let's take a look at some examples of probing diffusion within nanochannels. So substrates can diffuse into a nanochannel depending on how large that channel is versus the size of the particle that's going in. So if this is similar in size to the channel itself, then you're expecting it can only go through one by one. This is called single file diffusion. But if the channel is large compared to the size of your substrate that's going through, then as it diffuses through, the materials can pass each other as they go through. So this is called normal diffusion as they go through. We're about set to look at, so we've looked at both A and B, to look at contacts between the walls of the channel and the materials that are going through. So this example here with the little red dots are just some sort of contact, maybe a hydrogen bond or halogen bond, or polar interactions that might have some interaction with the guest or narrow the channel as the guest flows through. So we're testing effects of alternating diameters and interior functionality on diffusion. So one way we've done this is look at xenon diffusion in collaboration with groups at the University of Florida. And so our small channel, this phenylethylamine, is a little less than 5 angstroms in diameter. And you can fit xenon in there, but which you do under high pressure, but it gets distorted, kind of football shaped as it passes through. This distortion of that xenon is detected in the line shape of the xenon hyperpolarized 129 NMR. Versus our larger channel, the xenons can flow past each other. And so this shows normal diffusion. Here's an example of using our homogeneous materials to develop some new techniques. So we're looking at this in collaboration with the Great Tech group, and they are developing some fluorescence anisotropy measurements. So they took advantage of our one-dimensional architecture and high homogeneity to measure the excitation and emission polarization for this small bithiaphene dye. And so we equilibrated the thiaphene dye into our channels. And then what they do is they use a rotatable polarizer so they can excite the guest um, in the crystals. And so if the excitation wavelength is polarized in the direction of the fiber axis, we see a nice emission. But if it's polarized perpendicular to the axis, we don't see any emission. So this is an easy method to probe guest confinements in these low dimensional environments. Our group has done a lot of looking at reactions in confined environments. And so here's some of the hosts that we've looked at reactions inside. What we do is we crystallize these. So you heat them up in a suitable solvent like DMSO or DMF. And as you slow cool them, these crystallize, and then they have the solvent of crystallization in the channels. And so what we need to do first is we need to heat them to take that guest out of the channel and activate our host. And then we can put in a new guest. And we can either do this by soaking the activated crystals in a liquid guest or in a solution that has the guest in it, or you can load the new guest from the vapor phase. And these amazingly fill the channels to full capacity, even though it can only get in through the two ends. And we have experimental measures too that help us test this. So whether you have really large crystals and you load them, or you grind the crystals into like a 2-micron powder, you still get the same host-guest ratio. And that experimentally suggests that the guests only load into the crystals. How do we follow the absorption of guests? There's a couple ways we can do this. If you have a guest that has a nice chromophore, you can make a solution of it. And then when you put your activated host into the solution, the guest leaves solution and goes into the crystal. And we can see this from UV spectroscopy. We can see the depletion of the coumarin from solution. We can use a Beer-Lammer's plot and back calculate how much guest went into the crystal. Sometimes these host-guest complexes diffract, and I'll show you some examples of that. Other times they don't, and we get other information by PET or X-ray diffraction to look at the new complexes. So we've used this method to do reactions. We've done 2+2 reactions of enones. These load quite nicely into this phenyl ether host. And if you shine light on them, you get very high conversion and high selectivity for just one product when there are multiple products that can form in solution. And our computations suggest the reason for this is that the reactants are loaded in a specific way, geometry, close together. And that that allows or facilitates the formation of just one product versus in solution, they're tumbling and they can meet in many different ways and they'll get multiple products. We can do this in larger systems. So we've done coumarins and cumines and other materials. And if you load these into the larger crystals, we also get moderate conversion and high selectivity for some of the photodimers. In this one case, you can see this 7-methoxy. We don't get any conversion. And when we look at the modeling of that guest inside there, the two reactants, the neighboring materials, are loaded such that their 2 double bonds are very far apart. And so they can't undergo photoreaction in that channel. So it's the loading of the guest in the channel and how they're oriented with respect to their neighbor that controls the photoreactions that can take place. We don't have to do just photodimerizations. We can do reactions between different molecules. One that we're very interested in is using singlet oxygen as a reactant. So singlet oxygen is the simplest oxidizing agent, and it's generated using a sensitizer, a triplet sensitizer in solution, because ground state oxygen is triplet. And so if you irradiate the sensitizer, here are the sensitizers within our macrocycle itself. We can see formation of single oxygen here, and that's characterized by its emission at 1270. So we've used this to, as a way, encapsulation as a way to control the reactivity of single oxygen. So if you just used benzophenone by itself in oxygenated acetonitrile, you would get relatively good conversion, 68%, but you'd get several different products. You'd get the secondary allylic alcohol, the tertiary allylic alcohol, and reaction with benzophenone itself. If you use a benzophenone assembled bisurea macrocycle, and you put the reactant in the channels, so now you have a crystalline host-gas complex that you irradiate just in an oxygen atmosphere. In 2 hours, we see a higher conversion. We see none of the products that we'd see for benzophenone in solution, but we see something that's occurred within the channel. We see a primary allylic alcohol and a little bit of its overoxidation to the aldehyde. So that's quite exciting, of controlling the reactivity of single oxygen within a complex. My student Bailey was really interested in trying to understand what reactive oxygen species were being produced. And so she chose an unselective substrate. This one gives many different products depending on the reactive oxygen species that's generated. And so for benzophenone just in solution, you get many products, including these secondary allylic alcohol, epoxide products, and the tertiary allylic alcohol. But if you do the host, if you have the material encapsulated inside our host as a host gas complex, and you irradiate that in an oxygen atmosphere, we see really high conversion. So this suggests that oxygen can indeed enter and move rapidly through the solid. And you see reactions that are quite unusual. We see some of this enone formed that probably is formed through this intermediate as well. Our hypothesis is that we do generate singlet oxygen in the straws, and in that confined environment, the ene reaction that would take place also stabilizes this dioxirane derivative. And so that one is known, that can go on to give this enone as well as this tertiary allylic alcohol. So what we'd like to do is to understand what reactive oxygen species we're generating, superoxide control, that we only generate one, and look up how the different assemblies of our benzophenone carbonyl change the oxidation products that we see. And so one of my students, Raul Prakash, is looking at using these as catalysts in solution to try to do selective reactions with oxygen. So I've just given you a flavor of what our group does. We're really interested in the idea of how assembly modulates properties. And so here's an example from the Werthner group. They were looking at assembly of these diamides and how this modulates its photophysics. We're also interested in, modulating photophysics. We were looking at reactions of the benzophenone oxidations. In that process, we found out that one of the things we do generate are persistent radicals in these solids in low amounts. And so those radicals are actually quite useful. They can be used to enhance NMR using a special post sequence. Most of our current work is looking at understanding how the organization of triphenylamines might change and influence their photophysical properties. Triphenylamines are really of great interest to the polymer community because they can form these stable radical cations in solution and in the solid state. And so they're of interest as magnetic materials or polymeric magnetic materials. They typically need substitution to be stable. And so most triphenylamines you'll see in the literature have substituents at the pair positions out there along the triphenylamines. This helps slow degradation reactions and helps stabilize the triphenylamine radical cation. The triphenylamines are really good Hull transporters, and they can be used as one electron oxidants. And so you can generate their triphenylamine radical cations a couple of ways. One is through chemical or electrochemical oxidations. You can also do this through photoinduced oxidations. And so you can see that they're of quite interest as Hull transporters in solar cells. This is an example of using a triphenylamine derivative from the Docampo group as a thin layer in a perovskite-based solar cell. So our question is, how does assembly modulate these properties, the TPA properties? And we've made a series of compounds that we're reporting now that have the triphenylamines kind of constrained within a macrocyclic bisurea. And so we've made the ones that have the pair position either as a hydrogen, fluorine, bromine, or an iodine. I'll talk mostly about the bromine derivatives today. Obviously, triphenylamines are much more stable if you block the pair positions. And so we have some controls that have all the pair positions blocked. Here's the ones that have 4 iodines in it, so that will block all the pair positions. Here's one that just has a triphenylamine with the 2 pair positions blocked as with iodines, and the urea to look at the assembly. And so we're looking at how assembly could maybe stabilize these systems. One measure to see how stable they make it is looking at the stability of these systems if one of those positions are unblocked. If assembly can stabilize these radicals, then even things that have unblocked pair positions may be useful. And that would be nice for many applications, because if you have these steric blocking groups, that decreases the interactions these unpaired electrons can have with their neighboring group or with their environment. So ones that are unblocked may be of great interest in terms of their applications. So we have a couple questions. Can assembly stabilize these usually unstable radicals? Will these materials give photogenerating radicals? And are these useful functional materials? So kind of at the end, we're going to be looking at using light to do that electron transfer from the framework of the TPA to an electron accepting guest to see if we can make charge separated materials. So how do these assemble? Well, we see the urea is a great organizing motif. And so here's the bisurea with the 2 pair positions on each TPA unblocked. And these assemble very nicely into crystalline materials through the urea interaction. You can see if you're looking down the axis of the ureas, we have a cruciform pattern of the TPAs, organizing the TPAs in the crystal. Here's the crystalline material obtained by slow evaporation of this chloro derivative from acetonitrile. It also assembles by the urea interaction. The TPAs, though, if you look down that axis of the ureas, are more X-shaped. Here's the bromo derivative. Again, we have this nice urea interaction. This one is also X-shaped, isostructural to the chloro derivative. And then here's our last one with the iodides here. This one adopts a different structure. So if you look at one of the monomers, here's the monomer in pink, it's U-shaped. Okay. It assembles by the urea hydrogen bonds, but this is a little bit longer, so they're weaker because there's also additional iodine pi interactions, so halogen bonding interactions that help organize this into the crystalline structure. Our control materials also assemble. We expect these to give more stable radicals. So here's the tetraiodo. It assembles into columnar crystals kind of surprisingly through the urea interactions. You can see there's this nice open channel here that has, depending on what we've crystallized it for, either ethyl acetate or methanol inside. And here's another control compound that has the urea handle, but just one TPA on it. And so these assemble, again, by the urea interactions, and they have also additional interactions that bring these materials together. We can also look at the assembly of the macrocycles. These assemble as designed into the columnar structures held together by the urea interactions. This one assembled from DMSO, where we vapor fused dimethoxyethane into the crystals, and that's what's found in the channels. They give a lot larger crystals, though, than our other derivatives. And one reason for that, so they're about 10 times larger in size, is not only do they have this nice urea hydrogen bonding interaction, and so this is seen by a Hirschfeld analysis of our crystal structures. This red area shows where there's very strong interactions that are less than the sum of the van der Waals. So that's the ureas chains, but there's also interactions between the iodine and the aryl systems of the neighboring columns. And so there's pi-pi and halogen pi interactions in these materials, bromine aryl interactions. So we've made a whole series of materials with TPAs, and now we're going to look at their photophysics. So I'm sure you are just kind of a reminder for those of you who don't do much with photophysics. We're looking at the excitation of our TPAs. They can do, once they're excited, they can do several things. They can just relax and come back down to the ground state, so without giving off energy, or they could give off energy as they come down to the ground state, so that's fluorescence. They can do some charge transfer, right? So give away that electron that's in the TPA nitrogen, and so we could get CT states. That could happen from the singlet excited state, or you could do intersystem crossing to go to the triplet excited state, which could also give a CT state. From the triplet, you can though come back down. This is a slower process, so that's phosphorescence. As we have different halogens in our material, this should enhance the probability of this intersystem crossing, so enhance this process. And that's related to the coupling of the electron spin and the so spin orbit coupling. The heavy atoms that we have can be right on the molecule, so they could be covalent, that we've seen with our iodines or chlorines, or they can be things that we put in the channel, so they can be supramolecular as well. Okay, so we're going to shine light in it. How do we know where to shine light? Well, this is a solid state diffuse reflectance of our crystalline materials. And you can see they absorb here in this, 360, 370 nanometer range. This is quite different from solution. So for a solution, they absorb in around 300. So what we're going to do is we're going to irradiate these with 365 LEDs. And then we're going to look for radical formations. So unpaired electrons we can detect nicely by expand EPR. Okay, so do we generate radicals? So of course, if they're TPAs, we should generate radicals. So our first experiment was just looking at them in solution. So if you shine light on these, you can run over to the EPR and you can see, does this have radicals in it? You can see before irradiation, there's no radicals, there's no signal. But after irradiation, you can see this nice signal. So this suggests we're creating radicals. Okay. We can then run down to the NMR, right? Because this, of course, has on-block sites here that, make these unstable. And, in the 10 minutes between running from the EPR down to the NMR, you can see we've totally degraded our sample. So here's the black one, pre-UV irradiation, the red lines, the red spectra is post-UV irradiation. So these are not stable in solution. How about when they're assembled? So here are the nice crystals. When we UV irradiate them, they turn kind of a little coffee color. This is after an hour of UV irradiation. And if we run over to the EPR, you can see our nice signal that we're generating radicals. And we can then dissolve these up and go down and check the NMR. And you can see these are nicely stable. So assembly of these materials stabilizes things that are even unsubstituted. How about the columnar materials? So this is our assembled bisuria macrocycle. And again, no radicals before UV irradiation, but we can UV irradiate these and we get a really broad axial powder pattern signal that suggests we're forming radicals. You can do some simulations with easy spin, and to simulate a spectra like this requires two independent radicals, one with hyperfine splitting to the nitrogen. And so this suggests that we are generating two radicals. Our hypothesis, of course, is a radical cation and a radical anion. These materials still diffract after you irradiated them. And so we can go and take their X-ray structure, single crystal X-ray structure, and we see no changes in the X-ray before and after UV irradiation.
Dr. Shimizu, I'm sorry to stop you, but we have a question in the chat. Have you ever considered trying to use these porous channels in PISA systems to mimic something like ion channels in cellular membranes?
Oh, that's a cool idea. Yes, we did have some proposals out to look at membrane transport in these. We also, now that we can make these in higher yields, are looking at trying to attach these to kind of so the channels are approximately, these are approximately 6 angstroms in diameter. So there would be really tiny channels in terms of transport. But we are looking at integrating these with polymeric membranes.
All right, thank you.
So NMR and X-ray can't tell us if there's, less than 5% of something else there, right? So we're looking at, how much radicals are formed and, how much materials there are. We can look at the stability of these by solid state NMR. And so we've monitored the radical formation by cross-polarized magic angle spinning C13 NMR. And you can see for both of these systems, before and after UV irradiation, we see similar signals, but there's some broadening. right? Particularly for the linear analog, we see, which makes more radicals, we see broadening about the aromatic and carbonyl lines. And that's expected if there's a radical, right, that's in that area. So the methylenes and the urea bridge isn't as affected by the radical formation. And so this suggests that the radicals are not in that area. You can look at how stable these are by solid state NMR as well. And so after irradiating them to maximum radical amounts, we just stored them in the dark and acquired spectra for every 2 hours for 16 hours. And you can see these overlay perfectly. They're pretty much identical within the noise, demonstrating the stability of these materials over that long range. How many radicals are formed? Well, we can get an idea, an estimation of this by double integrating the EPR spectra that we get and comparing it to a standard. So we've done EPR of standard solutions of Magic Blue to make a calibration plot. And then we look at our materials. So these are the dimer structures, okay, either with hydrogen, so lots of en bloc pair positions, chlorine, bromine, and iodine in those positions. And you can see all of them generate radicals, but the bromine generates the most radicals, so 0.42%. It also, those radicals stay around a long time, so it has a half-life of 7 days. The hydrogen version, so this one assembled, you'd expect it to be really unstable, but these are very stable in the solid state assembled, and these do generate radicals as well, about 0.22%, with a half-life a little bit lower, 3 days. The iodine version generates much less radical, 0.06%, and they don't remain around, so their half-life is less than a day. So what are these? We've done some transient absorption spectra with the help of the Guzman-Tier group at Cincinnati. And you can see after radiation with the laser pulse, we do get this absorption here at 650. Our DFT, time-dependent DFT, suggests this is the radical cation. And actually bulk electrolysis also supports this conclusion that it's the radical cation that's formed. In terms of its sensitivity to oxygen, the lifetime of this material is not sensitive to oxygen, so that also supports radical cation, because if this was a triplet excited state, it would be quite sensitive to oxygen. So now we're going to go do things with these materials. So I told you these generate larger crystals, about 10 times the size of our typical ones. We can heat these and the crystals still diffract. And so when we take their single crystal structure, we see the same framework. The only thing that's missing is the electron density of the guest. And so what we do is we take our activated materials and we can expose them, immerse them in liquid guests. And so these are single crystal structures of complexes that have either benzene or fluorobenzene, chlorobenzene, bromobenzene, or iodobenzene. And you can see from their structures, the framework is unchanged. All the frameworks are isostructural. The only thing that's changing is the guest that's been put inside. So benzene, fluorobenzene, chlorobenzene, bromobenzene, all are absorbed 2 to 1, basically, close to guest. So I've shown you, and we've looked at some of the radical formation in those host-guest complexes. Well, what we really would like to do is to look at guests where we know would accept that electron. So we have a framework that's got this TPA, so electron-rich nitrogen that can, give that electron away to a guest, a good electron acceptor. So what have we chosen? Well, we've looked at ones that would fit into this interior. And so we've looked at benzothiodiazole. So its reduction potential is 1.5, dichlorobenzoquinone, and iodine. And we can load these in from the vacuum structures. And so you can see we'll have our host crystals here, the activated host. The guests will heat up so that they can become vapor, right? And they can load into the crystalline materials. Iodine, though, loads very fast. It's got a nice vapor pressure at room temperature. And so, in fact, it only takes about 2 minutes to load in to 2-to-1 into these crystalline materials. So, interest of time, Miguel. So here are the structures. Again, we have our isostructural host framework, so it's the same in all 3 materials. And we have our benzothiodiazole loaded inside, our electron acceptors loaded inside, and iodine. Iodine loads in really quickly, but once you take it out of the iodine vapor, it actually desorbs. So instead of being 2-to-1, already by the time we took it to the X-ray diffractometer, it's already started to come out of the material. So how about, do we transfer that electron? So let's take a look at our diffuse reflectance spectra. You can see here in blue, we have the activated host, and here's the crystalline material of the host guest complex. And you can see there's a shift here from the free guest and the host. So we have 2. And then if you shine light on these materials and then take the diffuse reflectance again, you can see formation of this broad band here, a charge transfer band at 486 nanometers. In the benzoquinone system, it looks like we have charge transfer prior to UV irradiation. So you can see here's our activated host, and black is that host guest complex, but it already has that charge transfer band at, this one's at 630, suggesting that there's charge transfer prior to, or spontaneous transfer of that electron prior to UV irradiation. We've also, it's like almost out of time, so I'll try to wrap up quick. We've also looked at this bicyclic voltammograms. So we've used CV to explore the redox behavior of these complexes. You can see here's the host. It shows 2 irreversible oxidation waves, 2 reversible oxidation waves at 1.46 and 1.86, and an irreversible one here near minus 0.46. That's similar to the TPAs. But in the complex, we retain those 2 oxidation waves, but we no longer see an irreversible oxidation wave near minus 0.6 volts. Instead, we see a reversible wave that minus 1 that corresponds to the benzothiodiazole. So this also suggests that we're getting that host guest, that charge transfer complex. So let's kind of wrap up here. I hope I've shown you that we can use assembly to stabilize less substituted triphenylamines. So it's another way besides using steric blocking groups. And we have materials now that have channels that we can put in, depending on the redox potential, guests that can accept that electron. So we've put in a benzoquinone that actually undergoes spontaneous electron transfer to give these host guest complexes, charged complexes. And we put in benzothiodiazole, which requires light. So a photo induced electron transfer to get that charge transfer complex. And in conclusion, self-assembly really enables a wider range of TPA structures that may be useful for as magnetic materials and in solar cells. Our future work, we're trying to look at these materials to understand the electron transfer process using advanced DPR techniques. We're also measuring conductivity for these materials to try to understand if these radicals that we generate do they represent mobile electrons or are they trapped states? And although we don't yet have the single crystal structures, we've been able to load things like pyrrole and thiophene in these materials and look at their polymerizations in the channels. So most of the work on the linear analogs was done by graduate student Mohammed Hossein, and on the TPA macrocycles was done by Faisal Islam, and of course Dr. Amin Sint, who started off these TPA projects. I'd like to thank NSF for their support of the BISURIAs and the Made in South Carolina grant from the NSF for helping us look at some machine learning that I didn't get a chance to talk about. And thank you so much for your attention. I'd be happy to answer questions.
All right. Well, thank you very much, Dr. Shimizu, for that great talk. And yeah, we're ready for some questions. I believe we have time for just a few if anybody has any. We have a question. What are the size limitations for the pore size you can achieve in these assembled macrocycles?
That's an interesting question. So we looked at maybe making tetraurea macrocycles, but they are too flexible. And so for the bisurea macrocycles, the largest we've gotten is up to 9 angstroms. I'm not sure if that's a maximum though. So I think if you have other weak interactions like halogen bonding interactions, that maybe we could go much larger. But some of the other larger structures, they might have different kind of our Purital pi interactions that, nature doesn't like too large a hole. So these materials are never gonna compete with moss in terms of like crazy large pore sizes, but I think they give a complementary way of looking at materials.
All right. Are there any other questions? Last chance. All right. Well, I think that's about it. Thank you again, Professor Shimizu, for the great talk. And we'll go ahead and hand it over to our next speaker.
Thank you again. So next up will be Professor Perry from UMass Amherst, and Hazel Davis will be chairing her session.
Thanks, Sophia. Good morning, everyone. My name is Hazel Davis, and I'm currently the president of the UMass PMSE Poly chapter here at UMass Amherst. It's my great pleasure to introduce Dr. Sarah Perry, a professor in the Chemical Engineering Department here at UMass. Dr. Perry's research group utilizes self-assembly, molecular design, and microfluidic technologies in order to mimic biological microenvironments. She recently received a 3M non-tenured faculty award and has been honored a number of times by the UMass College of Engineering for outstanding teaching, as well as a faculty advocate for diversity. I think I would also be remiss if I didn't mention when she's not revolutionizing how we think about complex coacervates. She's competing in world martial arts tournaments, as she was this weekend. And with that, I'll turn it over to Dr. Perry.
All right, thank you, Hazel. And so I'll apologize for my voice because, as she said, I was at a tournament this last weekend and I've been yelling and judging for the last 5 days. So it's a little bit rough right now. But anyway, thank you for coming to my talk. I'm going to change gears and look at still a type of supramolecular assembly, but one that's really done involving oppositely charged polyelectrolytes and how we can engineer these types of materials to create interesting products. So a lot of our inspiration in my group comes from biology. And so, if you were in biology class in high school, you probably had to learn a bunch of different organelles, but we largely traditionally have thought of these as being containers bounded by a lipid bilayer, right? So the nucleus, the mitochondria, the ER, the Golgi, et cetera. And in the last decade or so, there's been a real explosion of discovery and study of what people are terming membraneless organelles or biomolecular condensates. And so rather than being bounded by a lipid bilayer, these are actually liquid droplets that are phase separated inside of the cell. And the majority of these are formed by some interactions between intrinsically disordered proteins and usually RNA. And so I've got some examples here. The one that you have heard of but maybe didn't know it was a liquid is actually the nucleolus. But the ones that really have inspired us are ones typically related to stress. So these types of stress granules can form when you put the cell under, say, a metabolic stress, for instance. And what's really cool about them is that they can selectively sequester the specific enzymes associated with that metabolic stress, some of the RNAs as well. And so this raises a bunch of interesting questions related to how could we purify different biomolecules, or potentially other things. And what I'll highlight here is, what about vaccines and therapeutics? Right? We've heard a ton about the temperature stability issues of the COVID vaccine, but this isn't a new problem. People who have taken biologics, so a lot of the antibody treatments for like rheumatoid arthritis or other diseases, have really had to struggle with the temperature sensitivity of their medicines. And so the sort of standing questions here are, what are the underlying physics that allow us to control these systems? And then once we understand those design rules, can we then design analogous materials? Okay, so the strategy that my group has adopted in this space is using polyelectrolyte complexation. So what I'm talking about here is an associative phase separation between oppositely charged macroions. This could be charged polymers, proteins, surfactant micelles, virus particles, nanoparticles, et cetera. And if you imagine this situation where you have oppositely charged materials coming together, you could probably imagine some different situations, right? Maybe you have liquid-liquid phase separation. Maybe you have soluble complexes that are just nanometer scale dispersions. And so the two that I'm going to highlight here, if we have liquid-liquid phase separation, I'm going to term this complex coacervation. And so the micrograph that you're seeing here, it looks like an oil and water emulsion, but really this is an aqueous two-phase separation. You have polymer-rich coacervate droplets surrounded by a polymer-poor supernatant, and the entire thing is water-based. Now, you may or may not have ever heard of a coacervate, but you've encountered them regularly throughout your entire lives. So historically, these have been used extensively in the food and personal care products industries. It's how conditioning shampoo works. It's how things like fat-free salad dressing can attempt to have the same kind of mouthfeel associated with an oil emulsion, but without having the fat in there. But also in the research space, there's been a ton of work lately in terms of using these materials for drug delivery, underwater adhesives, and other things. And so it's possible to get liquid phase separation, but it's also possible and actually very common to get solid precipitation. And that's what I'm showing here on the right. And these types of solid materials historically were considered undesirable and intractable. And this comes from the perspective where if we're used to processing as engineers and chemists polymers, right, you would normally form some sort of a material by either working from a polymer melt, so using temperature, or solvent casting. And these polyelectrolyte complex solids don't respond to either of those. But it's also interesting that you can get both liquids and solids from the same set of materials just by changing the solution conditions. And so I'll give one biological example where we would like to understand this to avoid this problem. This micrograph here is work coming out of Tony Hyman and Simon Alberti's labs, and they're studying some of these intrinsically disordered proteins related to various neurodegenerative diseases. In particular, the protein they're working with here is associated with ALS. So that's the disease that Stephen Hawking suffered from. And the protein, wild type protein, non-disease causing protein on its own, will phase separate in a test tube, form really nice droplets. But if you have the disease mutant, what happens is those droplets form. And then over time, they age into this kind of spiny sea urchin-like granule that you can see. And the thought is that this solid to liquid transition actually is relevant for how the disease progresses. And so trying to understand. So this is an example where we would like to avoid solidification. But on the other hand, if we're trying to make materials, maybe we want that. Maybe we want to work with a liquid precursor, maybe a liquid coacervate, and then process it into more of a physical material. In this case, I'm showing you an example of some non-woven fibers. Okay, so that's sort of the setting. How should we think about coacervates? Well, for starters, they're really easy to make. This is literally a dump and stir preparation. So you would mix solutions of your oppositely charged polymers together. A lot of the work I'm going to show you today is going to take advantage of polypeptides because they represent a really nice platform for doing polymer chemistry where you can have explicit control over the sequence using techniques like solid phase peptide synthesis. And then because it's a peptide, you're basically keeping the polymer backbone the same and just swapping out the side chain chemistry. So you would mix together your solutions of your oppositely charged polymers, maybe with some salt. And what you'll see is your solution turn cloudy pretty much right away. And that cloudiness is due to the presence of phase separation. In this case, I'm showing you some nice droplets. So if this is what's going on, kind of what are the obvious parameters to look at? Well, it's electrostatic in nature. Probably one of the most natural ones is to think about the relative amounts of polycation and polyanion. And so we do a lot of this general characterization using turbidity as just the amount of light scattered from the sample, similar to optical density. And so this isn't a quantitative strategy, but it gives us a sense of where phase separation occurs. And in this case, I'm showing you a plot of turbidity versus the mole fraction of the polycation. And this is on a monomer basis. And so what you see is a peak around 50-50, right? Equal amounts of the two polymers. And that makes good intuitive sense. You want to be able to have your oppositely charged species pair up. Now, from an engineering perspective, though, this appears to be a really narrow window over which we're seeing phase separation. And so the good news is that these data were taken in the absence of added salt. And if you work at more physiologically relevant levels of saline, you see coacervation over a much broader range of stoichiometries. Now, I'd like to emphasize that if you look at the bulk coacervate material, if you isolate it and collect it, there's no difference in that coacervate composition wherever you are on this plot. What you're doing is you're effectively disproportionating the amount of polymer that you have. So if you have an excess of polyanion, in this case, at low mole fractions, right? The excess polyanion is just going to be in the supernatant. And whichever polymers are able to pair up will do so and form a coacervate. Now, there are some implications for this if you're working with more dispersions and trying to create colloidally stable dispersions, because you'll get excess charge buildup on the surface, but the bulk material is the same. And so since a lot of what we're doing is looking at kind of the bulk materials and the underlying science, we're going to perform all of our experiments here at this 50-50 charge fraction, simply because it's going to give us the biggest yield. Okay, so salt makes a difference. We could look at that a little bit more carefully. And in this plot, I'm showing you turbidity now versus the amount of added sodium chloride in this case. And what we see is that a little bit of salt seems to be good, right? Some charge screening going on, maybe allows the polymers to interact a little bit more. It's difficult to say from a turbidity plot whether you have more yield, right? But you're getting more signal. And so the way that we normally look at these plots is not to look so much at the details of the curve, but we'll look at, okay, there's coacervate formation. And then as we go to higher and higher salt concentration, we're going to reach a point where we no longer see phase separation. And that's the point of interest. And we're going to call that the salt resistance. And it corresponds to a point on the actual phase diagram. Now, what's interesting, though, is we can now use this as a thermodynamically relevant parameter to compare between different polymer systems. And so the data that I'm showing here come from this polymer polypeptide system, but I could compare it instead to a set of vinyl polymers, polyallylamine and polyacrylic acid. And so you see there's a huge difference in the response. Right? The polypeptides have a salt resistance, somewhere between 0.5 and 1 molar, whereas these vinyl polymers are stable up to probably 5 molar at least, right? We're pushing some solubility issues here. And then, how do we explain this? Well, you could look at these vinyl polymers and say, well, they're all aliphatic, right? There's no heteroatoms, so they're more hydrophobic. So therefore, they're going to want to stay phase separated because they're trying to stay away from water. And that would make sense. It's a great argument. Totally agree. You could also look at this and say, well, the charge density of these vinyl polymers is also higher, right? This is a 2-atom backbone. Peptides have a 3-atom backbone. Higher charge density, stronger electrostatics. Yeah, sure. Absolutely. And so this plot here represents an example of how it's difficult to really parse out the effects of individual chemical species when you change chemistry. And so to get around this particular challenge, I teamed up with my colleague, Professor Charles Singh at the University of Illinois, and his former PhD student, Tyler Lytle. And instead of trying to change the chemistry per se, we said, why don't we look at charge patterning? So here we're going to take advantage of the fact that we can synthesize sequence-controlled polypeptides. And we're going to then form coacervates between a polycation that is half charged and is sequence-controlled, and then a fully charged polyanide. And what chemically is going on here is we have copolymers of lysine and glycine, and then a polyglutamate. And we'll do the exact same experiment that I just showed you. So all of this work was done by my former PhD student, Li Wei Chang, and John Velez, who's undergraduate in my lab. And they would do those same turbidity experiments. Here I'm actually showing you the optical micrographs where, we're looking to see at what point do we no longer see coacervation. Record that number. And then we can repeat this for a series of copolymers that all have the same overall composition, but have different patterns of charge. And we're going to describe those patterns with a parameter that we've termed tau. And it's just the number of monomers in a repeat unit. So an alternating copolymer is plus 1 positive, 1 neutral. So it has a tau of 2. If you have 2 positive and 2 neutral, it's a tau of 4, and so on and so forth. And if I convert this micrograph image to something more quantitative, what you can see here is that the salt resistance increases with increasing charge block size. And again, if we're talking about electrostatic intuition, that probably makes good sense. You say, okay, I have this bigger run of charges, they're going to interact and then be less sensitive to, say, thermal fluctuations. Sure. We can repeat. What's cool, though, is that, so my lab does experiments, and Charles's lab works in the theory and computational space. And so we can now compare the experimental data from my lab to these predicted phase diagrams from the Singh lab, and we see the same kinds of trends. So how do we compare here? The salt resistance experiments that I showed you effectively walk along a line of constant polymer concentration, right? So any sample prepared under this 2-phase envelope will split, but you don't know anything about the compositions because you have the polymer pore supernatant arm of the binodal and then the coacervate. But the point at which you go from having phase separation to no more phase separation corresponds to a spot on this envelope. And so you can see the nice ordering in rainbow colors from red to purple here, increasing block size, increasing salt resistance, matches our data. We can also do an experiment where we change the concentration of polymer we do these experiments at, which would be like walking along this curve. And again, we see the same trends. Now, I'm saying we see the same trends, the parameterization we ended up with also got us to a point where we were fairly close in terms of near quantitative agreement, but I don't think we need to look into those numbers too closely. The important take-home message here, and proteins do this all the time, is that we can use sequence, chemical sequence, to change the behavior of our materials without changing composition. And so that's a very simple and powerful statement, but what's really much more interesting is the why that underlies it. And so this is where the simulation work that Charles's lab did really came in to help explain things. So this is just a schematic representation. And you would think that, like, okay, well, these are materials that form a complex. So probably those interacting materials, the final product, is going to be what dictates the phase behavior, right? And it turns out that's not actually the case. The important consideration is how those polymers are in solution before they ever see an oppositely charged species. So what am I talking about here? We know that you have to have charge neutrality. So a polymer in solution is going to have oppositely charged counterions, small molecule ions present in order to neutralize it. And if you have a relatively small pattern, tau equals 4, it's not a big deal whether this counterion is right on top of its monomer or if it's a little bit off to the side, right? Radially, that's pretty much the same. This is in contrast to one of these larger, blockier patterns where, because all of the charges are clustered in one spot, that's where you expect to see the counterions, right? There's going to be a much bigger electrostatic penalty to have a counterion sort of here in the middle of this neutral group. And the consequence of this is that when coacervation occurs, right, you have oppositely charged polymers coming together, we no longer need these counterions. And they can be freed up into solution. And the release of those counterions represents a huge entropic driving force. That's actually what tends to dominate the driving force for coacervation. And so if this release happens, the end state is the same, right? The entropy of the end state is the same. But if you look at this, there are a lot more degrees of freedom for these ions on these more smaller charge patterns than on these blockier patterns. And so the initial entropic states of these two polymers are different. And that's what gives you the difference in the phase behavior. So this is schematic. You can look at it with actual data from the simulations where you have the number of counterions as a function of chain index. And for little patterns, you see small oscillations, but then you start seeing these huge swings in the number of counterions. And so then you can take these data from simulation, map them onto an absorption isotherm and get entropy data out. And we can compare that experimentally with data collected from isothermal titration calorimetry. And we see very strong entropic driving force, as is expected. This is minus T delta S. It's a function of block size that increases with increasing block size. And again, we have near quantitative agreement between experimental and simulation results. So this is cool because it tells us it's the patterns ahead of time that matter, not the end state. And so we can get carried away, right? We can do all different kinds of patterns, try and understand, what are some of the subtleties going on. I'm not going to dig into this too heavily, but suffice it to say, a lot of your intuition will still hold, right? If this is a graph of salt resistance versus charge fraction, the higher the amount of charge you have on a polymer, the higher the salt resistance, right? That makes good intuitive sense. But what becomes more complicated is when you now talk about an interplay between charge fraction and sequence. And so I want to focus on these three guys right here, D, I, and F. And so if we look at these, they all have about the same salt resistance. D has the lowest charge fraction, but you can see here it has the largest blockiness, right? And then F similarly has the highest charge fraction and the lowest blockiness, and I is in between. And so there's a lot of subtleties and games that you can play, now understanding how this counterion condensation happens to sort of create the responsive material that you want given performance constraints and potentially composition constraints. Okay, so that's what I want to say about sequence. But there's obviously more than just sequence in the world, right? We can change polymer chemistry. And with the idea of getting towards more of a materials aspect, we switched away from polymers, and this is work done by my former student, Dr. Yatlin Liu, to look at the effect of just backbone chemistry. So we can make these polymers in gram scale quantities. The synthesis was done in collaboration with Todd Emmerich's lab here at UMass and Brent Summerlin's lab at the University of Florida. And the goal was to say, well, what's the difference of whether or not I have a methyl group on my backbone or not? And so Yatlin went and mapped out the full phase diagram, not just salt resistance, but here is the supernatant and coacervate arms of the binodal. And this is for our methacrylate-based polymers. And what you see is an increase in the width of the two-phase region with increasing chain length. And that's generally expected from theory. And then she did some other interesting things. Well, what if the polymers aren't the same length? What if they're mismatched? What's going on there? And so we did this comparison. So you can have both a short polycation and a short polyanion, and the data look identical. And since the polymers are basically the same, otherwise that, again, to me, makes good intuitive sense. But what was really neat is what if we put these data on top of the ones for the length matched systems? And you say, well, if they're mismatched in length, it's probably going to be a weighted average, right? Turns out that's not true. The phase diagrams for these length mismatched systems fall almost exactly on the same line as our shortest polymer system. And so why is that? Well, if we think about driving force, I talked so far about the release of counterions, but I haven't said anything about the polymers, right? Two polymers are going to have to come together to interact. That means they are giving up some, translational degrees of freedom. But if they're the same length, it just is what it is, and it sort of cancels itself out. We can neglect it relative to the driving force from the counterions. In this case, though, because we have a polymer that is so much shorter one than the other, there's order of magnitude difference here. Now, if I need to form a charge neutral complex, right, I can have one long polymer, degree of polymerization 500, and it's going to take 10 shorter guys of 50 length to neutralize it. Now the entropic contributions of those individual polymer chains become important. And then I can also get to this question that I started with of what about polymer chemistry? And so here I'm comparing same chain length methacrylate versus acrylyl coacervates. And you can see a huge difference in the size of the two-phase region, far more dramatic than anything we did with length. And the argument that I would put forth here is that the methacrylate polymers, right, they have a methyl backbone on their backbone. You could think of them as being more hydrophobic, right? They are going to form a denser, more polymer-rich phase, excluding water, because that's favorable to them. And so that I would argue is the difference here between these two systems. Now, this is all thermodynamics and phase behavior. What does it mean for the real world? What if you're actually handling a material? Well, we can look at this with rheology. And what people tend to do for coacervate systems is a time temperature or a time salt superposition. So you can take data at different salt concentrations. It's similar to time temperature because salt will soften the interactions. And you can create these master curves. And what this allows us to do then is compare samples at any different reference conditions. So what I'm showing you here, the vertical axis, I've shifted these data just for clarity. But you can see where this is all relative to the same as prepared salt concentration. And so as I go from my longest polymer to my shortest polymer, if we use the crossover in the two moduli as sort of a reference point, you see a shift. Shorter the polymer, it's a higher frequency, right? That means they relax faster. Makes good sense. But what the questions that come up then are, well, is this just a length effect, right? You change the length of the polymers here. What about concentration? I just showed you phase diagrams. Those are at very different polymer concentrations. And so then we can take advantage of this superposition, and instead of shifting them relative to the same as prepared salt concentration, we can shift them relative to the same measured concentration of polymer in the coacervate phase. And now you can see that they line up much more closely. There's still some small difference as you go shorter and shorter, that's probably a length effect. But this says that the majority of the effect in terms of how these parameters affect the rheology really has to do with polymer concentration. And we can ask the same question with regards to polymer chemistry, where we saw an even bigger difference in polymer concentration. So if I show you the data, and here you can actually see the individual data sets that went into this superposition for the same length methacrylyl and acrylyl polymers, just looking at the data, you say those two curves don't look the same. Right here we have a crossover in the two moduli. I don't see that here. Even if you've never seen rheology data before, you're like, those don't look the same, right? And in order to get similar rheological response for our acrylyl polymers, we had to go an order of magnitude up in length. And we think that this is exactly the same sort of polymer concentration effect that I talked about before. And so if you're talking engineering rules of thumb, you could argue that in this case, for these polymers, adding that methyl group, it gives you about the same thing as increasing the chain by a factor of 10. Okay. Now, I had motivated this whole talk with the idea of, well, couldn't we mimic biology and maybe stabilize proteins? And so let's come back to that. Now, the question, though, is why do I think that coacervates would be useful for this? Well, for starters, inside the cell, right, things are compartmentalized. Let's keep incompatible things apart. Okay, so coacervates are phase separated, they can do that. But more importantly, if you think about what the environment is in a vial of protein, right, solution of protein on the shelf, it's a very dilute system, aqueous, maybe a little bit of salt. The inside of a cell is extremely crowded. This is a snapshot from a molecular dynamics simulation of the inside of an E. coli cell. But this, it's like 30% by weight, polymer, protein, RNA, however you want to define it. So there's not a lot of space for things to potentially unfold and denature. But what I think is important really in terms of the compartmentalization and how things form is you want some physical crowding, but you also, cells have evolved the sort of soft interactions, kind of the right, interpersonal interactions between the different molecules to help stabilize things. And so can we get at this to harness what's going on? All right. So if I'm talking about putting a protein in, this complicates the system. And so this is all work done by my former student, Dr. Whitney Blocker-McTighe, where she kept polymers the same, two polymers, changing the ratio of the two polymers, and then added in a constant level of protein across the entire system. And so just for comparison, this black data is very similar to the turbidity data I showed you before. And if I add in bovine serum albumin, which is negatively charged, you see a shift in the turbidity peak. And that makes intuitively good sense, right? You threw in something that is negatively charged, you're probably going to need a little bit more polycation to neutralize the entire system. And then we can still form nice coacervates. But looking beyond just their turbidity, we want to know where the protein is. We want to quantify this. And so she can separate out the two phases and quantify the amount of protein in both the supernatant and the coacervate. And so if we start off, our sample has about 50 micrograms per milliliter of protein. If we don't see phase separation, say at the ends, right, we recover pretty much all of that protein in our supernatant. But once we start having phase separation, you see a depletion of protein from the supernatant that's matched with a commensurate increase in protein in the coacervate. Now, this is a little subtle, but I have two axes here, right? I started with 50 micrograms per milliliter, and I ended up in the coacervate with 2 milligrams per milliliter. So you get a huge concentration effect by doing this. And so we can take those maximum levels, and I'm going to map them out. We can talk about a partition coefficient, so the concentration in the coacervate phase divided by the supernatant, or encapsulation efficiency. I'll mostly talk about partitioning because it's thermodynamically relevant, and we can do this for different proteins, right? So if BSA has an acidic isoelectric point, what that means is that as I increase in pH, I'm going to increase the net charge of the protein. It's going to get more and more negative. And as we do that, we see a commensurate increase in the partitioning. Again, intuition's working for us. The same thing will work if you take a cationic protein like lysozyme. In this case, though, you're going to be going down in pH in order to increase the charge. But now let's do a side-by-side comparison here, right? BSA has a partition coefficient of 30, which is nothing to shake a stick at. But lysozyme is pushing upwards of 1,000. And so what's the difference here? And so we tried to look at, what's the difference in the charges on the proteins. And if I just look at a colored structure of a protein, I can't tell you if there's clustering of charges or not. So we took a more analytical approach and did a pair correlation analysis. So we took the three-dimensional structure of the protein, identified the spatial locations of each of the charged residues, and took each one as a reference. So I start here, and I go out into space, and I say, where's the next charge residue? Record this, normalize it, etc. And so that's what you're seeing here is this plot of the pair correlation as a function of distance for each of the charged residues. And for lysozyme, it's pretty, but there's not a whole lot to see. Or for BSA, I'm sorry, there isn't a whole lot to see. But for lysozyme, you see these really sharp peaks, right? G of R of 2 or 3 at short distances. And that tells you that there's a cluster of charges, in this case, 3 lysines right here. And that's interesting because the presence of that charge is relevant to lysozyme. It's a substrate binding pocket. And then for BSA is a serum protein. It's present in large concentrations in your body. It's probably good that there aren't sticky patches for it to glom onto things. And so this tells us that if we know something about our protein, we could have expectations in terms of the charge, the net charge and the charge patterning of how it'll go into our coacervates. But these are pretty different proteins, so we could do a slightly better control experiment here. So let's compare BSA with hemoglobin, right? They're about the same size. If you look at their pair correlations, they're not patchy at all, but there's a big difference in charge, right? BSA is far more charged than hemoglobin. And so we would expect that it would have better uptake. And that's exactly what we see here, right? Far higher partition. You can do the same thing comparing hemoglobin then to chymotrypsin, right? So chymotrypsin is a little bit smaller, but it has about the same net charge and a little bit of patterning or a little bit of patchiness. And so same charge, more patchiness. Yep. Chymotrypsin falls right in between the 2 other proteins. So we're developing these rules of thumb and these, kind of heuristics for understanding this. I think in the interest of time, I'm going to skip this in terms of patchiness of the polymers, but your intuition will hold, right? If you have a highly charged polymer that's competing with your protein, it's going to affect your ability to encapsulate it. What's more interesting, though, is the idea that you could stabilize these proteins. So here, I talked about proteins, but I'm actually going to talk about stabilization of viruses. This is work that we did with Karen Held's lab, in particular Shuai Mi, a former student at Michigan Tech. And we're using some animal viruses as veterinary analogs for human diseases. Now, the encapsulation works the same way, right? You can do your same turbidity experiments. But then when you go to quantify the amount of virus that's present, you don't quantify virus, or we didn't choose to quantify virus in terms of a concentration. You use it, you use an infectivity assay. So we're only able to detect infectious virus. And that's interesting because we got this weird double peak in our turbidity, and we're like, did somebody fall asleep in the middle of the experiment? Like, we never see a double peak. But it turns out there's 2 different populations. It's a heterogeneous mixture of probably unformed capsids or empty capsids and infectious virus. And we were able to selectively fractionate out the infectious virus. But this was the most exciting result, where this is for a non-enveloped porcine parvovirus. And we're comparing just the parvovirus in solution with the parvovirus in our coacervate. And the y-axis here is an activity titer. So it's a log scale titer of infectivity in certain cell line. And then we exposed these samples to a week at 60 degrees. And so, yeah, maybe we did lose a little bit of viral activity by putting the viruses in our coacervate, but you see we lose a lot less over time. And so this, I'm not saying this is good enough to solve vaccine issues, but it's a really exciting first result. And so we're trying to push this further. In the last couple of minutes, I just want to comment about using these coacervates as materials, right? I talked about this solid to liquid transition. In all of these phase diagrams I've shown you, there's no theoretical prediction for solid materials. But it turns out that you end up with solid materials, usually at low salt, and it's a kinetically trapped effect. And you can see that here is if I go from high salt to low, it kind of looks like I go from a liquid to more of a gel, and now I've expelled more and more solvent. And this phenomenon is exactly a gelation phenomenon, where you can think of the electrostatic interactions that hold the coacervate together. As you pull salt out, this salt ions would allow these chains to rearrange, and without that salt, they'll become basically trapped and unable to rearrange, forming a physical gel. And so you can test this out rheologically, but this appears to be a general phenomenon for coacervate materials. But it also means that you can use salt concentration as a handle for processing things. And so this was a collaboration that Zoe Meng did. She was a jointly advised PhD student between my lab and Jessica Schiffman, where she took that coacervate material and used it to electrospin polymer fibers. And this was really cool because we could do this from a purely aqueous solution, no organic solvents needed. And you can see here the optical or the SEM micrograph of the resulting fibers. They look a little bit rough, and that's the salt coming out onto the fiber surface as they form. Now, why is this useful? Well, being able to do fully aqueous processing is important for a number of reasons, depending on what you're trying to do, right? From an environmental standpoint, if you can decrease the amount of organic solvents, that's always nice, right? But what are the cargoes you're trying to encapsulate, right? If you were, say, trying to encapsulate proteins, they tend to prefer an all aqueous environment. And so potentially we could make, here I'm showing, these are just dye-containing fibers, but you could have fibers containing enzymes, that are immobilized for some sort of a reaction, other types of things, and maintain their activity. And so we think that there's a lot you can do with this. What's also interesting, at the beginning of the talk, I told you that solid precipitates had traditionally been considered to be intractable because they didn't respond to temperature or organic solvent. Well, that bug in the system is actually a feature for us, right? I can form fibers. You can see the salt crystals. I can rinse them, have the salt go away. And then Zoe went and got solvent, all samples of all the solvents we could find in all of the polymer science labs and put fibers in it and walked away for 2 months. And you can see that the fibers persisted in every single sample, right? Trifluoroethanol is a really great solvent. It dissolves a lot of things. Yeah, after 2 months, it was a little squishy, but you can still see that there are fibers there. This particular polymer set are strong polyelectrolytes, so they're pH, they're not responsive to pH. But if you wanted to, say, recycle your system, you can recycle them by adding high concentrations of salt. And so this represents an orthogonal recycling strategy. If you were to, say, create a layered laminate with like a coacervate type material and a more traditional organic solvent soluble polymer. And so lastly, there's another cool feature here. So electrospinning, you have this, the electric field is pulling this fiber jet. And in order to keep that jet from breaking up into small droplets, right, Rayleigh instability, you typically have to have the polymers be long enough that they're physically entangled, so that as the jet's moving, the polymers don't have time to relax and allow for droplet breakup. And so this has meant that electrospinning has specifically required very long polymers at very high concentration. And that can be described in terms of, this is a plot of polymer weight percent and molecular weight. If you have just the overlap concentration where the sort of polymers are interacting but not overlapping, about 10 times above that is the entanglement concentration. So the idea here is that the rule of thumb is you have to be 4 times above the entanglement concentration to get fibers to form. Well, we can do this study with a bunch of different length polymers, right? Long ones, short ones. Is that even a polymer? And it really is this question of, long spaghetti versus rice or penne. And what was super exciting is that instead of having to have physical entanglements, the associative electrostatic interactions that drive coacervation are actually sufficient to allow us to form continuous fibers, even with these really short oligomers. And so this allows us to decouple processing requirements, in this case for electrospinning, with other design criteria that we might have that would be length dependent, right? So maybe you need your fibers to dissolve after a while, or other parameters. So that was the story that I wanted to tell. We try very hard to couple sort of fundamental studies of self-assembly and phase behavior, things like charge patterning, chemistry, et cetera, length, to understand how we can make effective functional materials. And so I've just got some examples here of the fibers. Here are these colorful coacervates and the resulting fibers. But ultimately, all of this work comes down to questions of controlling the interactions. How do the molecules interact? And then what are the right design rules to say, in this case, create a coacervate that could help to stabilize a therapeutic protein? So I've tried to acknowledge my collaborators as I've gone through. My lab loves doing collaborative research. So if you have some cool ideas, please let me know. There have been a ton of people that we work with here at UMass. My longtime collaborator, Charles Singh at Illinois, and all of his group. Synthetic chemists, and Karen Held for her help with all of the virus work. And then here's my group doing the best that they can to have fun in these pandemic times, along with other key members who have become more and more important as they crash Zoom calls and seminars, et cetera. And with that, I'd be happy to take any questions that you have.
Thank you so much, Dr. Perry. Really fantastic talk. So we have a couple of questions in the chat. So the first one is, for your polylysine and polyglutamate studies, what pH were your solutions, as they are weak electrolytes? And have you done a pH study in the context of coacervate stability?
Absolutely. Yeah, so all of the studies that I showed you here for the polypeptides were done at neutral pH. So we estimate the pKa for polyglutamate is around 4.5. For lysine, it's like 9.2. And so we generally try and be at least 2 pH units. If we're not doing a specific pH study, we try and be at least 2 pH units away from the pKa and then equidistant, so that we can assume in that case, if you're 2 pH units away, that, yeah, to a first approximation, you're fully charged. Now, having said that, right, so these calculations, or these data agree with the idea that my system is fully charged. If we had done a pH study, say if I'd raised the pH to the pKa of polylysine, I don't have the data in this talk, but what happens is these curves shift. So if your polycation is half charged, right, now it's going to take 2 of them for every 1 polyanion. And then the way I have this graph defined here is fraction. So you would expect this peak to shift from 50-50 to 67%. And that's exactly what you see. And we've looked at this. There's some interesting other challenges that the Terrell group at the University of Chicago has shown going down in pH, because you can get hydrogen bonding with some of these carboxylic acids, but that's a secondary complication.
Awesome, thank you. Another question with respect to the spun fibers that have salts on them. So you're washing off these salts with water and other solvents. How does that affect the stability of the fibers?
Yeah, so we don't think that washing the salt out really, it depends on what you mean by the stability and also the mechanical responsiveness of those fibers. If we assume that our solid materials have no salt in them, right, whether there is crystalline salt on the surface or not should not affect their properties. If you did have the salt still present in the solution, right, then if you were to put this sample back into water, right? You're going to have that extra salt there that will then add to whatever you put in to affect, the dissolution in that case. But our hypothesis, and we're trying to test this right now, is that the concentration of salt that you prepare things at shouldn't affect the mechanical properties. Or if it does, then we have some cool things we have to think about once it's a solid.
Awesome, thank you. And then I think maybe we'll have time for just one last question. If you could comment on the effect of molecular weight influencing specifically the salt resistance of these macerates.
Mm-hmm. Yeah, so if I pull up here, the molecular weight effect is more important on the small side, right? As you go longer and longer, you sort of attenuate and affect things. And so it doesn't show well here in terms of salt resistance because of the way that this is graphed. But if you just looked here on the polymer rich side of things, the trends will be the same. As I go up in molecular weight, I go up in stability with salt. And you would see the same thing, but this particular graph isn't going to show it on the low salt side.
Awesome. Well, thank you so much. Really great talking. We appreciate having you here today.
All right, thank you.
Thank you again, Professor Shimizu and Professor Perry, for wonderful talks and starting off our August series. So again, this is the last month of the seminar series. We will meet again in 1 month, in 1 week, and we will be hearing from Professor Ann McNeil and Professor Michael Scholes. So if you could join us again next Tuesday, we look forward to seeing you. Have a great afternoon, everyone.